Hi, everyone. Thanks for joining my talk today. I hope you've been enjoying the series and the several talks that I've been having. I believe this is the 10th talk in my Science and Religion series, and this is on molecular machines. Now, you might be asking yourself, what do you mean by molecular machines? Kind of see a picture here, but not exactly sure. Well, I'll explain. So first, let's talk about the idea of a black box. And so a black box is a system or machine that someone finds interesting, but they don't know exactly how it works. Now, this may not be relevant for you, but for some, people know, okay, how to use a computer in the sense of they can type on the keyboard, they can play games, they can maybe make a resume on Word, but they really don't know exactly how the computer is making all of those things happen. So for them, the computer would be an example of a black box. They might know a little bit about it, but they don't know the details of how it actually works. In Darwin's time, the cell was a black box. They could see it, they kind of knew a few things about it, but they truly didn't know how it actually worked. They speculated that it was actually due to electricity, is how the cell was able to work and function, and they actually thought the cell was very simple. However, um, it's very complicated and it's run by a lot of micro machines, and a few we'll talk about. So Darwin, in his Origin of Species, when he postulated the idea of evolution and natural selection, he actually also said that there was a test. And so, with his idea, he provided a test to kind of determine if if evolution or if natural selection could be like debunked. So, how would we know what would we be able to find that would say, okay, that this isn't really possible, natural selection isn't possible? And so Darwin's test, what he wrote in The Origin of Species, he said if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, then my theory, meaning Darwin's theory, would absolutely break down. So what he was describing there is he said if you could find anything that was considered, the term we use nowadays is irreducibly complex. And so if you could find an irreducibly complex system, then that would say that, okay, that is an example of something that would then break down natural selection. So irreducibly complex is a system or device that has a number of different components, a bunch of different parts that all work together to accomplish a task that the system is designed to accomplish. And if you were to remove even one of those components, the system would no longer function. And so, what this would mean is this type of system is highly unlikely to be built kind of piece by piece, step by step through Darwinian processes, through natural selection. So then it would have had to have been developed in another way, which is against what evolution and natural selection says, that everything was formed in that process. So if we found something that couldn't be formed in that process and was formed in another way, that would then kind of break down his theory. So let me provide you an example of something that's considered irreducibly complex. So here's an example of an irreducibly complex system. So a simple example is a mousetrap. It's not a biological example. I'll give you biological examples later, but just something to kind of begin with. So a mousetrap has several parts. It has this wooden platform right here. It has the metal hammer, which is kind of the little bar that kind of falls over and traps the mouse, squeezes the mouse. Then you have the spring, you have the release catch, which is the part that when the mouse steps on it, it's what releases the hammer. And then you kind of have this metal connector bar that connects the catch to the hammer. That is kind of the trigger that releases that hammer. Now, if you take away any of these parts, then it's not like the mousetrap just works 50% as efficiently, or, oh, it might only, trap 50 out of 100 mice. It just doesn't work at all. So if you take any one of these parts away, now the mousetrap just doesn't work. So not only do you just need these 5 parts, but you also need them to be matched to each other in a kind of a spatial relationship. So it's not that you just need the spring, the hammer, the release catch, the platform, and the bar, but you need them to be in the right way. If the spring were in a different position, and it wasn't next to attached to the hammer, then the mousetrap wouldn't function the way it is. So we need all the parts and we need them to be laid out in the correct spatial relationship to each other. So this is an example of how irreducibly complex systems defy Darwinian explanation, because you can't just all of a sudden have something that is really complicated. So how natural selection works is that there's slight successive modifications. So it doesn't jump from something simple to something very complex all of a sudden. It slowly adapts and changes something to get an increased complexity, is what natural selection says. And so we can't just all of a sudden have something very complicated. And in this case, we wouldn't be able to have just slight modifications, because if it's missing a part, it wouldn't work. And if something doesn't work, then it's not going to continue on with natural selection. So if there is an adaptation that changes and now it's not functional, then that species, that whatever it is, would not continue to exist, because with natural selection, only the things that kind of make the species more fit or make it more efficient, make it work better, is going to continue on. Now, there is a counter argument to the mousetrap that was produced by Kenneth Miller. And so Kenneth Miller said, okay, well, what if we say, well, okay, so maybe it won't be a mousetrap anymore, but it still might be useful. So if I were to take away 2 parts, so for example, the catch and the metal bar, then we might not have a mousetrap anymore, but we would have a 3-part machine that would kind of be like a tie clip or a paper clip. So for example, here he's saying if I take away the catch and the metal bar, so if I take away the catch here and this metal bar, then you just have the spring and the hammer and the platform. So it'll kind of be like this, which would be like a paper clip or like a tie clip. Now, if you then took away the spring, then you just have a 2-part system, like a keychain, because then you would just have the hammer as the metal part attached then to the wooden platform. So the point which science has long understood is that we can have bits and pieces of supposedly irreducibly complex systems, and that they may have different but useful functions. The question for evolution, though, is not whether you can take a mousetrap and use its parts for something else. It's whether you can start with something else and make it into a mousetrap. So, what the argument of Kenneth Miller is, let's have the endpoint and let's try to work backwards. Like, if we just take it away, is it useful? But that's not what evolution is trying to actually say. Evolution is saying we can start from one part and then we can assemble this mousetrap. And so that's the issue that evolution has to address, is whether we can start with these parts and then make it into a mousetrap. So you still have the problem of how would the mousetrap be assembled? And not only would we have, how would we get these parts, but also how would we get these parts to be spatially in the same area? And this is the problem that evolutionists don't really address. Now, obviously, they don't sit around and talk about the mousetrap, but when we talk about some biological examples, they don't talk about how we would actually get to the end product. So it's not like we could just take the end product and back it up. Now you may think, oh, well, maybe if I took the end product and tried to take its parts, maybe then I could see how those parts came together. And that would be a good idea of where evolutionists could start. So if we were trying to say, like the mousetrap, if we could find the paper clip, for example, or the keyring in nature, and then you could start there and show how it formed a mousetrap, that would work. But that doesn't actually exist in real world examples. So let's actually talk about some real world examples. So let's talk about the cell. Okay, so in the cell, we have, or at the cellular level, there are a lot of biological machines. So actually, life is based on molecular machines. And so inside our bodies, we have these little molecular machines that do lots of different things. For example, they haul cargo from one place in the cell to another place in the cell, kind of transport it around. It also turns cellular switches on and off. So there's some switches that are kind of like gates that either tell the gate to be open or closed. So if we, turn that on, turn on or off that, we can adjust if the gates are open or closed when we're trying to have, different ions passing through or different things passing through the gates. We have pulleys and cables. There are electrical machines that let current flow through the nerves. We have solar powered machines that capture the energy from light and store it. And there are molecular machinery that lets cells move, reproduce, and process food. So there's a lot going in within our bodies that have kind of a machinery aspect to them. So I want to give you our first example of what is an example of an irreducibly complex system. So the first example is cilia. So cilia are kind of hairs, kind of like that are whip-like. So they're whip-like hairs on the surface of cells. And there's two different ways that they kind of interact. So the first would be if the cell is stationary. So you can kind of see this in this example. So if it is stationary, then the cilia here, they move fluid across the cell's surface. So they kind of flow and then they move things across. So here, the cilia, one example of it is that you have cilia that line your respiratory tract and they sweep mucus to your throat. And so they kind of allow and kind of move to kind of, they move so that they can move other things that may be in the way that the cell doesn't want there. However, if the cell is mobile, then the cilia can actually row the cell toward fluid. So it can be used in this kind of whip-like fashion to kind of move as like a propeller. And so an example of this would be sperm cells. So you can see here in the bottom right picture, sperm cells also have these kind of hair-like, whip-like hairs at the ends of them. And so these sperm cells are propelled forward by kind of the rowing action of cilia. And cilia is not a simple system. It actually contains 200 protein parts that allow the cilia to move. So it's a very complex system. So how does the cilia actually function? So I'm going to kind of break down this picture that's in the top right of your screen as to how the cilia are able to kind of have this kind of whip-like kind of motion. So there are 9 microtubules. These are long, thin, flexible rods, and so they're the little circles. They're the kind of red, almost look like 8s that are going around in the picture. And these microtubules encircle 2 single microtubules that are in the middle there. These outer microtubules are connected to each other by Nexon linkers. And so you can kind of see those with the little lines that are drawn there. Each microtubule has a motor protein, and that motor protein is called dynein. And each motor protein attaches to one microtubule and has an arm that reaches over and grabs the other one. And so what it kind of does is it reaches over and it kind of pushes it down. And so what happens is this one will kind of reach over and push it down and then push it down. And you kind of get like the 2 rods start to slide lengthwise with respect to each other like this, because it's kind of reaching over and pushing this one down, then this reaches over, pushes it down, and so you kind of get that motion that's going. So the 2 rods will start sliding lengthwise with respect to each other. And so what happens is these Nexon linkers, they're originally really loose, but then as they push, they're going to get, as it's pushing down, it's going to start to get kind of stretched out. And when it gets stretched out and taut, that's where we start to get this bending action. And so as the Darwinian pushes further, it starts to bend the apparatus because as it's trying to push down, you can even see with my hand, it's going to start to kind of bend. And so that's how we can get this bending or this kind of whip-like action with the cilia. So what do we need for the cilia to actually move? So like we talked about with the mousetrap, how there was those different parts. What are the different parts of the cilia? So for the cilia to move, we need those rods, we need the linkers, and then we need the motors that will allow this all to happen. It won't work if you're just missing one or two. Now, there are some examples of them that have less rods, but you can't work with no rods. So you can have examples with less rods, that kind of thing. But if you have no rods, then there's nothing for the linkers to link and there's nothing to be moving. So if you're just missing one part, it won't work. And then each part did not have a prior purpose in the cell. So some people have suggested that maybe the motor proteins that are used in cilia could have previously been used in something else. Maybe they were cargo proteins. But if they were cargo proteins, then they wouldn't be strong enough to actually move the rods. So cargo proteins have a very specific design and purpose, and so we couldn't just allocate them as motor proteins in our cilia. So we have to have all of the components. And not only do we need all the components, like I said with the mousetrap, they also need to come together at the right place and at the right time. So first we need them to exist, period. And then we need them to come together at the same time and in the right spatial relationship. So how can you kind of imagine how probable is that to happen? So I want you to kind of picture you're at a fair, at a county fair, and there's like 10,000 people there at the fair. And so I want you to link up. So I want you to be able to, I want you to, everybody else is wearing blindfolds, you're wearing a blindfold, and you're going to have to link up. So this is just three parts. And so what you're going to do is you're going to reach out and you're going to grab two people's hands in the middle of the fair. And I want you to try to find two people with your name. So in this situation, there are two other people. My name's Christina. So I would need to find two other people with the name Christina. And so I'm in the fair, I walk, I grab a hand and I grab a hand and I try to say, okay, are these two people Christina? And this is what I'm looking for to make my cilia in my example here. Okay. And oh no, this one isn't a Christina. So now I need to grab another hand. However, when it comes to the mutation rate in the cell, it's really, really slow. So what that means is the next time you would get to kind of try to grab hands again would take a very long time. And so what it would actually be is you would link hands and then you wouldn't get to try again, but one time in a year. That's how slow the mutation rate is. So if there's 10,000 people, it's going to take a very long time, an improbable amount of time, an unrealistic amount of time for me to be able to find the two other people by just randomly linking hands in with only getting to do it once a year. And so this is the idea of even if the three parts existed in the cell, for them to then be able to be in the same place at the right time is just not very realistic. And so that's what kind of makes the cilia to be a irreducibly complex system because it's not able to be produced by natural selection. And right now, evolutionists have not been able to provide any step-by-step explanation for where cilia originated from. So I'm going to give you a second example. This is a more popular example, and it was also shown at the beginning of my talk. And so this example is the bacterial flagellum. It is similar to cilia. Cilia acts like kind of oars when they row because of that moving action, whereas flagellum are like a rotary propeller. So kind of think about the back of a boat and this kind of propeller, rotary propeller on the back of a boat. And so the flagellum is actually a much more complex system than the cilia. So how does the flagella work? So the propeller is long and whip-like, so it's kind of got a similar look as the cilia, and it's made from the protein flagellin. And so that would be kind of this blue segment here in the picture in the bottom right. And the propeller is attached to the drive shaft by a hook protein. So you've got the drive shaft, you've got the propeller. We need something to kind of hook it as like a universal joint. And that would be kind of this, I guess, brownish looking color that is between the blue um, kind of propeller there and the filament, and then the pinkish purple looking color here, that would be the drive shaft. And so this joint actually allows the propeller and the drive shaft to rotate freely. So it's not a very fixed, kind of stuck in place joint. And then we have several proteins that act as bushing material to allow the drive shaft to penetrate the bacterial wall and attach to the rotary motor. So if you can see here, everything that's kind of on the outside of my picture here is the membrane wall, bacterial wall here. And so then we need to be able to get this into the membrane. And so we have several different proteins that are allowing all of this to be in there. So you've got the drive shaft that's right on the outer edge of our membrane, our bacterial wall, and then our rotor, which is down into deeper into the uh, bacterial wall. So kind of all the pinkish parts there are inside of the membrane wall. Then you have the hook protein that kind of comes, and then you have the flagellin that comes, the propeller made out of flagellin that comes after that. So those are all kind of the parts to this. So how does the bacterial flagellum actually get its energy? So its energy is generated by a flow of acid through the bacterial membrane, which that system itself is actually really complex. So even just the way that the bacterial flagellum gets its energy is a complex system in itself. And so through this flow of acid through the membrane, which is then going to be charged, we are able to get energy from that. Now, how well is this motor? How well is this propeller? You know, you may think of motors in your car, motors on a boat. How well do they run? So the propeller of the bacterial flagellum can spin at 10,000 revolutions per minute. So this is really, really fast. And not only can it spin very, very quickly, but it can stop spinning as it's spinning. It can stop spinning within a quarter turn and instantly start spinning the other way at the same speed, at that 10,000 revolutions per minute. And so, and not only is it a very fast, but it's also very efficient, being able to turn over one direction to the other very quickly. Now, we're used to thinking of kind of motors, engines, all of that as big objects. So if you're thinking your car or if you're thinking the propeller and motor for your boat, these are very big things. However, not only is it impressive to get something that would have kind of this power and this energy, period, in any kind of system, big or small, it's even more impressive to get it at a very small segment. If you think about it, we really haven't made anything engineering wise that even compares to this for like a boat motor. So how small is the flagellum? So the flagellum is only a couple of microns. And so depending on your system of choice, a micron is about 1/25,000th of an inch or 1/1000th of a millimeter. And actually, most of its length is that propeller. So the majority of the size of the flagellum is the propeller that's kind of sticking out. So the motor that's within the bacterial cell wall is only 1/100,000th of an inch or 1/4000th of a millimeter. So it's very, very small. So here in the top right, here are just some more diagrams of this. This is from an electron microscope, so you can see it as if we were just kind of taking a picture of it in the cell. So who's steering the bacterial cell? So this is helping. We've got the flagellum, but how is it actually like going? So we can see that it's going to spin and it's going to go really fast, but it could be spinning really fast, but like who's driving the boat is how you could think about this. Like we have a very powerful motor, but who's steering the boat? Is it just going to go off wherever? Well, it actually has complex sensory systems that feed into the bacterial flagellum, kind of turning it on and off and changing its direction. And so the way that you can kind of think about this is if you had a missile, so certain missiles, smart missiles, have guidance systems that kind of help them find their targets. And so the bacterial flagellum also kind of has a guidance system that directs to where it needs to go. So let's talk about, is this an irreducibly complex system? So for this to be able to evolve from Darwinian evolution, it would need the paddle, so the flagellum, the rotor, and the motor for it to work. Without one, it won't work. So you need the paddle, you need the rotor, and the motor. So those are the three parts that you would really need. And for this to even work, we need about 30 to 35 proteins to create a functional flagellum. And at this moment, no one has discovered a step-by-step evolutionary explanation. And even if we wanted to say, well, we could take the parts away, and if we were to find the parts, maybe we haven't found the process to go from the parts to the bacterial flagellum yet. Well, the parts aren't found anywhere else either. So we haven't even found any even hints or aspects of this so far in the cell or in other parts. So evolution does not have an explanation for this and for the complex design and system that it has. So this is another example of an irreducibly complex system. So the cilia, and we have the bacterial flagellum. But you might say, okay, well, these are kind of just like two examples of kind of motors or systems. So I want to provide you with a third example. And this example is going to be a little different because it's more of a system. And so I kind of hinted with the flagellum that the way it got its energy used a kind of complex system, and the guidance directions of where the flagellum goes also is complex system. But I want to give you an example of another system that is actually considered an irreducibly complex system. So this is the cellular transport system. And so we're going to be talking specifically about eukaryotic cells. So these are cells that are found in all organisms except for bacteria. And in eukaryotic cells, there's a bunch of different compartments. So you can kind of think about the cell as being a house. And in this house, there's a bunch of rooms. So for example, in the cell, you have the nucleus, you have the mitochondria, you have the endoplasmic reticulum, the Golgi apparatus, ribosomes. You have a bunch of different parts that make up the cell. And if we were to actually kind of go through and break it down and try to figure out like how many parts or sections or rooms it has, there are about over 20 different sections that are sealed off by membranes. So it's not only is it like a room in the sense of a part, like a segment, but it's actually even more like a room because it's sealed off. So it actually kind of has like walls how you can think about it. So it's not just like, oh, we can kind of like, we have an open floor plan and we're just kind of designating different parts of the house. No, these are actual true rooms in the sense that they are sealed off with these membranes. So that's why we can say that there are about over 20 different sections. Now, an interesting thing is that cells are constantly getting rid of old stuff and manufacturing new components. So they don't just like exist, make everything, get it all set up, and then just hang out. They're constantly, upgrading and making newer stuff and making sure everything's working well. And so these are designed to work in only one room, but not other rooms. So when they make a component, it's designed to be for this one room and nothing else. But all the stuff is kind of made in one central location. So you can kind of think about it as like the factory where everything's made. And that would be the ribosomes. So ribosomes are where everything is kind of made, centrally located. So where are the ribosomes? So if you can look at my picture here, so here's the cell. You have the rough endoplasmic reticulum, and the ribosomes are kind of like these little dots or little balls, however you want to think about them, on the endoplasmic reticulum. So the ribosomes are a collection of some 50 large molecules containing more than 1 million atoms. So even though they're very small, the cell is small, the endoplasmic reticulum is small inside the cell, and the ribosomes are small on that. They still contain a lot of molecules and atoms. And like I said, they're kind of like a factory. They can synthesize any protein that it's instructed to make by the DNA, including they can also make other ribosomes. So this is a really awesome factory because it can make anything that you ask it to make, and it can make another factory even. So with this transport system, what do we need? So we have the ribosome, we have the factory within the cell, but now I need to get everything out to each of the places. And remember, each component has a very specific room that it needs to go to. So let's think about this kind of system, this transport system that we need. So if I make something in the factory and I need to get it to this new place, I need some way to transport it. And so we have molecular trucks. These are enclosed and with motors, and they can carry the components. So that's one part of our cellular transport system. Now, we also need kind of a highway for them to travel along. How are they going to physically get from the ribosome to the new location where they need to go? Third, we need a system for the truck to identify which component to pick up. So if my truck, say I'm the truck in the real world, I'm the truck that comes to the factory to pick up bananas, and I'm going to take it to the store that is selling the bananas. Well, then I need to know where to go to make sure that I don't get computers in my car, in my truck. I need the bananas. So there's got to be some way for the ribosome to know what to put into the truck and for them to kind of communicate. So we need some sort of system there to make sure that the truck gets the right component that it is scheduled to deliver. So then the truck gets the component. It needs to know where to go. So even though the roads are there, we need some way for it to know where to go. So even though I might get my bananas, I need to know how to get to the store that's going to sell those bananas. So I need a system that's going to tell me where to go. And then once I get there, I need some way to unload. Because remember, the rooms that we talked about have those membranes. So they're enclosed. And so then we need something that's going to say, oh, we need our component that the truck has delivered. We need to open up so that the truck can actually unload that component into the compartment, into the room, and then the room can have what was delivered. So there's a lot of different parts here for this whole system to work, from getting something made in the ribosome all the way to getting it, transferred into the right room, into the right location in the cell that needs that part, that component. My apologies. And so this is a very complex system. So if you think about it, if we were to remove any part of this system, it wouldn't work. So if we were to no longer have highways, then how is the truck going to get from the factory to the place that it needs to drop it off? If the truck is just going to collect anything, then how's it going to know where it needs to go if it doesn't even know what it's collecting? So any one of these parts, if it is removed, the whole system will not work, will get stuck. And so this is another example of a very complex system that evolutionists have not been able to really explain how this works. And this is information. This isn't necessarily talking about how the ribosome is actually making the physical thing. This is just the information needed to kind of transfer. We're not talking about the information needed to make the molecular truck, to make each of these rooms. That is a whole, that adds to the complexity of the system. We're only talking about the transport system. So if we were to get into those details, it would actually make the entire system a lot more complex. And like I've said with the other two examples, this example as well, evolutionists have no explanation for kind of the stepwise process, because you can't just say, okay, well, we'll make the trucks. Well, what's the point in having trucks? They don't just make anything for no reason. Natural selection is making something for a purpose, like not for a purpose, sorry. Natural selection is making something, changing something, adapting something so that it will be more efficient, will be better for whatever the environment is. So it doesn't know what the end goal is. And so why would it make a truck if the truck isn't going to be useful because it doesn't have the rest of the parts? So this is another example of an irreducibly complex system. All right, the fourth example that I want to share with you is blood clotting. And so this is also kind of a system, but in this sense, it's a system that also has a physical result. And so that's just kind of another example of an irreducibly complex system. So for blood clotting to work, we need the blood to clot at the right place, in the right amount, and at the right time. If all three of these things do not happen, you end up bleeding to death. So it's really, really important. We need right place, right time, and right amount. And actually, this whole process of this happening, without getting into the particulars, involves 10 steps and about 20 different molecular components. It's a very complex system. But the difficult part with blood clotting isn't the clot itself. So creating the clot is complicated, but it's actually not the hardest part. The hardest part is regulation of the system. And so in the sense of right place, right amount, right time. That's the hard part. So for example, we need it to be in the right place. If we make a clot in the wrong place, say for example, you have a clot in your brain or a clot in your lungs, then you die automatically. We need it to be at the right time. If we make a clot 20 minutes after you have this cut and we need it to clot and it takes 20 minutes, all the blood is going to drain from your body and then you're going to die. And we need it in the right amount. And so if we have a cut, we need it to just clot right there at the cut, and we need it to just be a small amount that is associated with the size of that cut. So we need to make a clot that's not confined. If you make a cut that is not confined, my apologies. If you make a clot not confined to the cut, like to the cutted area, and it goes to your entire body, and if your entire blood system ends up clotting, then you would also die, because we need blood flow for oxygen and other processes. And so, like all the other examples that I've shared so far, there's no step-by-step process of how blood clotting was developed. We don't have an explanation of the cause of these steps, and it's actually not fully really understood the whole process. And so this just gives you another example of a complex system that not only is it information, but has a physical result that is irreducibly complex. So in summary, irreducible complexity demonstrates how these interconnected biological systems could not have been created through the numerous successive slight modifications that Darwinism demands. So remember I said at the beginning that Darwin actually stated this test for reliability of natural selection. So he said, here's my theory of natural selection. And the way that you could kind of break it down or disprove it is if you were to find something that was considered irreducibly complex. That was a system that could not be broken down. And so just now I demonstrated 4 different examples of irreducibly complex systems, but there are way more than just 4. There are hundreds of examples in biology of these irreducibly complex systems. Also, it shows an alternative. Irreducibly complex shows that, okay, natural selection is not the answer. There's actually an alternative. Because for us to have these complex biological machines, we need information. And who is the creator of information? Intelligence. And so this points to some sort of designer somebody that was intelligent that designed these aspects, these systems, and these machines. And so these irreducibly complex systems kind of give evidence to a transcendent creator or an intelligent designer. So I want to ask you, Charles Darwin conceded that his theory would absolutely break down if it could be shown that any complex organ could not possibly have been formed by numerous slight modifications. So now that I've showed you 4 examples, do you think that these 4 examples have passed Darwin's test or not? Do you agree or disagree? So that's something for you just to think about. And you're also welcome to Google and search for more examples of complex, irreducibly complex systems to see other examples or to even dive into these 4 examples into more depth. So thank you so much for attending my talk. Here are a few references that you may look up and to follow. And I hope you join me in my next talk. We'll be looking at the challenge and issues of DNA and how does DNA relate to natural selection and evolution.